Welcome to the next section of our series of videos on the cytoskeleton, and we're going to have 5 videos in this section on microfilaments. So let's get started with that. As you may recall, microfilaments are the smallest of the cytoskeletal filaments. When we say smallest, I mean the narrowest, approximately 7 nanometers in diameter, best known for their roles in muscle contraction. And we're not going to get into that because you'll deal with that if you take physiology. They're involved in things like cell migration, amoeboid movement, cytoplasmic streaming. They're important for the development and maintenance of cell shape due to the organization of actin into networks in the cell cortex just under the plasma membrane, and they're critical in the structure of microvilli. So looking at the slide here, microvilli have these bundles of actin filaments. The cell cortex is a mesh, gel-like network of loosely connected actin filaments. Adherens junctions can be connected via a belt of actin. Filopodia have a gel-like mesh of actin, as does the lamellipodium, or leading edge, of amoeboid or crawling cells. Important structural features are stress fibers that provide structural strength to cells. There's a role for the actin network in phagocytosis and also for moving around endocytic vesicles. And last but not least, you're going to see a ring of actin working with myosin to pinch the dividing cell in two through the formation of a structure called the contractile ring. Now in the diagram here, it looks like this ring is on the outside when it's actually just on the inside of the plasma membrane. So here are some microvilli. These are the kinds of villi that you find on the surface of intestinal cells, and inside those villi you have tightly packed bundles of actin, and then down here there's a looser network of gel-like actin. In this crawling cell here, we can see a network of actin just under the leading edge. We can see it in the filopodial spikes, and then we can see these stress fibers which act as providing tensile strength and anchoring the cell cytoplasm to the substrate when the cell is crawling. So let's now talk about the monomer used to build microfilaments. We talked about alpha and beta tubulin and microtubules, so let's talk about actin. Actin is probably the most abundant protein in eukaryotic cells. It's found in all eukaryotic cells. Once it is synthesized, it folds into a globular-shaped molecule that can bind either ATP, or if the ATP becomes hydrolyzed, ADP. So the G of G-actin stands for globular actin. The globular actin molecules polymerize into chains, so that would be an example of quaternary structure, to form the microfilament, which is 2 twisted helical filaments of polymerized G-actin. And so the polymerized G-actin is called F-actin or filamentous actin. When you look at the G-actin sequence, it's highly conserved within organisms and between species. There are some variations. You can basically divide the actin family up into muscle-specific actins called the alpha-actins, and then the non-muscle actins, beta and gamma actins. And if you look at beta and gamma actins in cells, they tend to localize to different regions. So the G-actin monomers can polymerize together into these individual filaments, and that's a reversible process. So we see a lag phase as polymerization initiates, and then we see an elongation phase, and there's also the ability to shorten as well. So the G-actin polymerizes into these filaments, and then 2 filaments wrap around one another to produce the F-actin or the microfilament form. All of the G-actin monomers in these filaments have the same orientation. That means they're all stacked up in the same way, like you can see here. There's just a slight twist, obviously, to each of these fibers, but you see the same orientation of each of the globular actin molecules. Actin filaments have a minus end and a plus end, but unlike with the microtubules, it's harder to tell which end is which. And so a technique called decoration can be done to show which end is minus and which end is plus. So to do this, you take a subfragment of a myosin, which is a motor protein, and you mix it with the microfilaments. And because of the shape of that subfragment of myosin, you get this kind of decoration of barbs, of kind of like arrowheads. And I've kind of drawn this in here in PowerPoint, so you can see there are these like arrowheads, and you can just about make out these arrowheads. And so the pointed end of the arrowhead points towards the minus end, and then the barbed end, which would be this end here of the arrowhead, is at the plus end. So you can use that to determine the plus and the minus ends. And so this polarity of the filament is reflected in more rapid addition or loss of G-actin at the plus end than the minus end. So we see more rapid addition of G-actin to the plus end than we do at the minus end. So once the actin with ATP bound adds to the plus end, there is the hydrolysis of the ATP to produce ADP. So a small region at the plus end of the microfilament has actin with ATP bound, but the majority of the filament has ADP bound. So it's a little bit like what we saw with the microtubules, where you have kind of like a cap of GTP bound tubulin on the plus end, except here it's ATP bound actin at the plus end of the microfilament. So that's all we're going to do to start with. We'll come back in the second part and keep looking at these microfilaments. I'll see you soon.